Hi, my name is Barbara Novak, and I will be talking to you today about using Nextflow to break computational barriers, or NASA's approach to democratizing space omics data. So I will start off by talking a little bit about space biology and what type of data we are processing. Then I will talk about NASA's Open Science Data Repository and our analysis working groups. Then I'll switch to talking about enabling fair science by both standardizing metadata and standardized data processing. And I will talk a little bit more about our data processing pipelines and workflows, how we measure the impact of our repository on the broader scientific community, and then next steps. So over the past 10 years, the amount of data, the number of objects that have been launched into space has increased dramatically, as you can see in the plot on the left. And that means that the amount of data that we've been generating has also dramatically increased. And so in the NASA Space Biology program, where this project is located, we focus on characterizing the different risks to living systems that come with environmental exposures encountered in space. We leverage data from a variety of different analyses and experiments, and I'll talk about how we're making these data publicly available, standardized, and curated. And so these pictures are a representation of some of the hazards for biology and health that organisms encounter when they leave Earth's orbit, such as microgravity and altered gravity, ionizing radiation, confinement, isolation, and hostile or closed environments, altered temperature and atmosphere, Martian and lunar regolith, and altered day-night cycles or circadian rhythms. It's also important to think about combinations of multiple stressors and the timeline of impacts. So is it something that will impact health immediately or over a longer period of time? So when you're thinking about gathering data about spaceflight, one thing to know about that is that it's really expensive and very difficult to get a large volume of data. So a single mission or a single organism provides a really limited view into how a biological system responds to spaceflight. So it's really important to be able to gather data across hundreds of studies or many organismal comparisons or across missions, assays, and data types to get a bit more context and be able to create multimodal insights. And that way we can get a more complete emerging puzzle of how spaceflight impacts biological systems, and we can get a better scientific readiness for crewed missions like the missions to send astronauts back to the moon or onto Mars. So NASA's Open Science Data Repository is a project to try to enable reuse of multimodal and multi-hierarchical fundamental space life science data to advance basic science, applied science, and operational outcomes for space exploration and knowledge discovery. So OSDR is composed of 4 separate projects. ALSDA, or the Ames Life Science Data Archive, is the official repository for non-human data generated by NASA's space biology program. GeneLab is the official repository for all omics data from non-human spaceflight. And NBISC and SMCC are repositories for actual physical samples. NBISC is the National Biological Institutional Scientific Collection and is a biorepository of non-human samples collected from NASA-funded missions and correlative ground studies, and SMCC is the Space Microbial Culture Collection. All of these projects come together in OSDR, which provides a single submission portal for the data, user interface and website tool for research data submission agreements, workspace for submitter labs and their collaborators, striving for maximally open access with necessary controls for sensitive data. So we try not to expose human data, for example, in any of the samples that we collect. Our data is maximally fair or findable, accessible, interoperable, and reusable. And we also strive for transparency, inclusivity, and reproducibility. So we work with the greater space biology community via our analysis working groups, or AWGs. At the moment, we have 12 AWGs listed on this slide, with over 1,000 members and 32 different subgroups that investigate all sorts of different things. The AWGs work together and with us to set data standards, both for metadata and for processing standards, as well as standards to enable reuse and data mining. They also collaborate with each other and sort of serve as a collaboration platform for scientists to work together. And two examples of that are the Cell Press Package from 2020 on the biology of spaceflight, and the Nature Portfolio package from last year on SOMA, the Space Omics and Medical Atlas. For a full list of OSDR and GeneLab enabled publications, you can see Google Scholar at the link below. Oh. And I forgot to mention that anyone who is at all interested in space biology can check out check out our AWGs at the link on this slide and fill out the form to join if you're interested. All you really need is an interest in space biology. So how do we enable fair science? How do we make data findable, accessible, interoperable, and reproducible? One very important component is standardizing the metadata. So we have built a metadata model called the ISA tab format, standing for investigation, sample, and assay. The investigation table includes all of the information about the study. So just an overview of what was done in the experiment. The sample table includes sample level information, including sample source and sample names, as well as characteristics and factors pertaining to single samples. The assay table then contains assay level information for each sample, including things like the technology type and the platform that was used to generate the data associated with those samples, links to the data files themselves, and protocol references, etc. All of the data associated with the sample is linked and downloadable. Everything uses standardized formats and standardized ontologies. All of that information is hosted on GitHub in the OSDR data curation repository. And so all of the originally submitted data comes in various files, multiple tables, non-standard characters, non-standard naming conventions, and our curators transform that into comma-separated files with single tables, standard nomenclature, standard units, no spaces, uniform statistics, to make that data standard and interoperable across studies. So the next step in enabling fair science is standardized data processing. And that's building consensus data processing pipelines with the scientific community. What we want to do is ensure that users of all skill types can access the data. So for complex data types, such as sequencing data, it can be quite difficult for biologists without an extensive computational background or access to compute resources to make use of the raw data. For these types of data, we work with our AWGs to build standardized pipelines and workflows to process the raw data into a final processed format that users can access on their own laptops. And here's an example of 4 of our standardized data processing pipelines: RNA sequencing, amplicon sequencing, metagenomics, methylseq. Once we have data in a standardized format, we also want to make it easy to find and view. So we've built several interactive search and visualization tools. First of all, there's just our data repository, which is a web front end for all of the data. Each study gets its own study page. And there are some simple filters and search options for finding data within the repository. For more complex queries and data mining, we also have a biological data API. And for searching and requesting our biospecimens in NBISC or SMCC, you can use this biospecimen search tool. The biological visualization portal provides a view into the data for data mining without needing to actually download the data and do it yourself. So we have some pre-canned visualizations for being able to compare dynamically across different studies and different assay types. Finally, our environmental data application and RadLab portals provide specialized visualization for environmental and radiation data, respectively. So now moving into pipelines, so we make a distinction between a pipeline and a workflow. For us, a pipeline is just the description of how GeneLab processes a specific omics data type hosted on OSDR. So here's an example of our RNA-seq processing pipeline for eukaryotes. All of our processing pipelines and associated documentation is available in a single GitHub repository at NASA/GeneLab data processing. Once we have pipelines defined and we've agreed with our community that that is how we should be processing the data, we implement those pipelines as Nextflow workflows. The documentation and source code is also publicly available in GitHub. At the moment, we're in a transition period where we are moving that workflow code out of that one main documentation directory at GeneLab data processing into separate repositories for each different assay workflow. So this image is a representation of our GeneLab RNA-seq workflow. And as you can see from it, in addition to the RNA-seq consensus pipeline, which is kind of in this gray box, our workflow adds an analysis staging workflow for pulling data in from OSDR and a validation and verification pipeline, which I will talk about a little bit later. We use these workflows internally to generate process data that we host in OSDR. All of our workflows use containerized and or packaged environments for reproducibility and flexibility. So every single workflow we have, we run internally using Singularity, but our workflows are also able to work with Docker out of the box. And many of our workflows are also able to work with Conda environments instead to provide the greatest flexibility for people who want to use our workflows themselves. So that validation and verification pipeline that I mentioned before, represented at the bottom of this workflow diagram, includes uses multi-QC for metric extraction as well as report generation. And so how do we measure the impact of creating this repository with all of this data, all of this process data? I can sort of share some numbers about how much data we have. Those are the numbers on the left in this slide. So 580 studies, over 263 terabytes of data. The pie charts show a breakdown of that data into different assay types, different organisms. And all of this data represents 122 original publications with data that was submitted to OSDR or pulled from other public data repositories such as SRA. There are also 130 peer-reviewed enabled publications. That is publications from analysis that was performed on data that was pulled out of OSDR. So data that had already been submitted before and is now being used to generate new insights and new research. Over time, you can see that the rate of growth has been faster for the enabled publications than for original publications, which shows that scientists have been able to perform new research with data in our data repository. So what's next? We would like to democratize access. So through a collaboration with the Los Alamos National Labs, GeneLab workflows are moving into the cloud via the NASA Edge Bioinformatics platform, which uses access compute resources underneath, allowing users who do not have access to their own compute resources to run our pipelines on their own data, or to reprocess OSDR data, perhaps with slightly different parameters than what we use internally. We have a proof of concept up at nasa.edgebioinformatics.org that is our amplicon sequencing workflow. And if you're interested in becoming a beta tester, you can sign up by filling out the form below. I would like to thank the Open Science for Life in Space teams at GeneLab, ALSDA, and BISC and SMCC, and all of the wonderful members of our Open Science Analysis Working Groups. We get our support from the NASA Space Biology Program, the Biological and Physical Sciences Division, the Science Mission Directorate, and the Human Research Program. Thank you very much.